Hello. In this video, I'll be presenting on the Module 11 journal article, Virtual Core Flow Cytometry by Swalwell et al. Published in 2009, which discusses an alternative method of cell analysis in flow cytometry. Flow cytometry is a technique used in many research fields such as oceanography, stem cell biology, and DNA analysis. This slide demonstrates a typical flow cytometry setup. On the left, you can see the cells of interest being inserted into a flow stream of sheath fluid exhibiting laminar flow. The cells are injected at a slightly higher flow rate such that the cells are pushed into the center of the stream, allowing only one cell to pass through the center of the flow cytometer at a time. As the cells pass through the center of the setup, they are hit with a laser beam, and the light is scattered from them is detected as shown in the top right figure. This light is split into two categories: forward scatter, which correlates to the size of the molecule, and side scatter, which correlates to the granularity. These two types of scatter are plotted against each other and create the flow cytometry plot, shown in the bottom right, where each cell is represented by a dot. Groups of cells can be generated to represent different cell types. The problem with this typical setup is that it requires a large amount of sheath fluid on the order of liters, which is impractical in field or low resource settings. In addition, if a sample ever needs to be retested, the concentration of cells in the fluid decreases drastically with every test and can be diluted up to 100 times the original concentration. To address this problem, the researchers built the system shown here based on confocal microscopy, using position-sensitive detectors made of dichroic mirror and 3 photomultiplier tubes named D1, D2, and FSC in this picture. A microscope focuses the light scattered from the individual cells and sends it through a pinhole, which is detected and evaluated for fluorescence measurements. Cells that are inside the quote-unquote virtual core will create sharp images, whereas cells outside of the virtual core will create blurred images, thus mimicking a typical flow cytometer. First, to test whether the position-sensitive detector could sufficiently detect movements of the cells in the virtual core versus the traditional flow cytometer, 1 micron and 6 micron fluorescent beads were injected into both systems, and then the stream was moved side to side by 25 micrometers. The left shows the schematic of what the graph should be when the light is only detected by FSC, shown in 2A, or by all 3 photomultipliers, as shown by 2B and 2C. The right graph shows the results of the side-to-side motion of the stream, which also displays the ellipse shape, indicating that the position-sensitive detector is calibrated. 2 test cell groups were used to compare the virtual flow versus the sheath flow. First, murine T cells, which were labeled with CD4 and CD8, were tested. The left graph shows the results using the traditional sheath fluid, where the ratio of cells expressing CD4 and CD8 was found to be 2.11. The middle graph shows the distribution of all cells inputted into the virtual core system, and the right graph shows the distribution of only the 1.4% of cells that were properly aligned in the system to take accurate fluorescence measurements. This right graph shows that the CD4 to CD8 ratio was found to be comparable to the flow cytometer measurements at 2.14, only a 2% difference than the traditional system. The next cell type test run was to compare traditional flow cytometry to the virtual core model to classify raw seawater based on physotherin and chlorophyll. Like the previous example, both graphs show similar grouping and ratio of cell groups present. So in conclusion, the virtual core model is comparable to traditional flow cytometry. This is a less complicated, less resource-intensive option for field labs or low-resource settings, and the system can be used to recharacterize samples without diluting them. The future directions would be to apply this system to more cell types and more situations, and retest samples to determine if the same plots are generated. In addition, it would be ideal to increase the percent of cells that move through the virtual core such that flow cytometry graphs generated can represent all cells in the sample.